Okay, here's another pretest for you. At what point in this action potential graph are the sodium channels open? If you answered right here, then you're correct. It's the opening of those sodium channels that causes the charge of that cell to become much more positive in this case, otherwise known as depolarizing. If we want to have a nerve impulse, then what we need to have is some sort of stimuli that is enough to change the electrical charge of a neuron. So both our cells and our neurons have an electrical charge to them. Inside a cell, we see a negative charge of about -5 to -100 millivolts. And in a neuron, we see a negative charge of about -40 to -75. Now at rest, a cell membrane of a neuron generally is going to have a negative electrical potential of about -70 millivolts. And this is an important number to commit to memory. So what's actually causing this difference in membrane potential? Well, it's largely due to the permeability of the plasma membrane to various ions, specifically ions like sodium, potassium, chloride, and calcium. Generally, what we see is that a neuron is going to have a high concentration of potassium ions, and this is abbreviated with a K+, on the inside of the membrane, and a high concentration of sodium ions, which is abbreviated with an Na+, on the outside of the membrane. It's the imbalance in number of the ions inside and outside of the cell membrane that causes this resting membrane potential of roughly 65 to 70 millivolts. Now we know the membrane can become permeable to both sodium and potassium, which are the two ions we're going to primarily focus on because they have the greatest influence. But we have to have something working in the background to maintain our resting membrane potential. And that thing working in the background is called a sodium-potassium pump. It functions to move 2 potassium ions into the cell and move 3 sodium ions out of the cell. And that's going to make a little bit more sense as we delve into how an action potential works. It's important to point out that sodium and potassium are not the only ions that influence the membrane potential. You also have the chloride and the calcium. But notice you actually don't have a ton of chloride and calcium either intracellularly or extracellularly. Instead, the ones that you have particularly on the inside of a cell is sodium and potassium. And so these tend to be the major players, and we know how they influence an action potential when a membrane is excited. And specifically notice that we have a high amount of sodium outside the cell, a high amount of potassium inside the cell. When that cell becomes excited enough for the cell to depolarize, you're going to get an influx of sodium into the cell through the channels that you see on the upper right-hand side. Now, at some point, we need to bring that membrane potential back down, and that's when the sodium-potassium pump is functioning in the background to help restore us to what would be a resting membrane potential. So pumping out those sodium ions and pumping back in the potassium ions. Assuming that you have a strong enough stimulus to cause a cell to depolarize, you're going to walk through a full action potential. And again, that process is an all or none process. So let's walk through on this graph exactly what's happening to cause that cell to depolarize, and then what happens after the cell has depolarized. In the very first step, what happens is if we have enough of a stimulus, so an excitatory postsynaptic potential is being delivered, then what we get is an increase in charge until we hit a threshold. That threshold is -55 millivolts. Once that threshold is hit, it will open the sodium channels and you will get a big rush of sodium into the cell. Remember, there's a high proportion of sodium out of the cell and a small proportion of sodium in the cell. So it wants to move down its concentration gradient and come into the cell. Because sodium has a positive charge, this is going to increase the overall charge within that cell. After a cell has been depolarized, if you want to have another action potential, the cell has to repolarize. In order to repolarize, the sodium channels are going to close. This is going to prevent any additional sodium from moving down its concentration gradient and coming into the cell. And instead, the cell will open potassium channels. Remember, we have a high concentration of potassium within the cell and a low concentration outside of the cell. So if we open additional potassium channels, it'll help move potassium out of the cell as it flows down its concentration gradient, thereby lowering the overall charge of our cell in this case. Now sometimes the cell overshoots and it takes the cell to negative, and we call this hyperpolarization. Luckily, we have our sodium-potassium pump that's working to pump 3 sodium out and 2 potassium in that helps equalize then our resting membrane potential around -70 millivolts. And as we've stated before, this is an all or none policy. So once that action potential is initiated, it's going to carry itself through. We also have something that's called an absolute refractory period and a relative refractory period. And what that's referring to is whether or not another action potential could occur if a strong enough stimulus were initiated. During the absolute refractory period, this is any time that the sodium channels are open. And during this time, there is no way that you can have another action potential occur. So again, absolute refractory period, anytime a sodium channel is open, no way you're going to get another action potential. But the relative refractory period is after the action potential, or rather the depolarization phase, when you're repolarizing. So your sodium channels are closed and your potassium channels are open. During this time, if there's a strong enough stimulus, again, it's going to have to be a stimulus that's stronger than what that initial stimuli was to cause the initial action potential. You can generate another action potential, but it's got to be a really strong stimulus in this case. So that's, again, our relative refractory period. Sometimes the graph in isolation is confusing for students because you have a hard time visualizing the sodium and the potassium ions. So this figure is just showing the same thing from the previous slides, but it allows you to see that actual movement of the sodium and potassium ions. So we have sodium in orange and we have potassium in pink on the inside of the cell. So initially we have a resting membrane potential, high sodium outside of the cell, and then a high potassium inside the cell. When the cell becomes depolarized, the sodium is going to start moving into the cell. So you see that those orange little ions are moving into the cell, and that's also our upslope in our action potential. Now during the repolarization, to try and lower the charge, because remember both of these ions have a positive charge, the body is trying to kick out, in this case, the potassium ions. And so it's opening the potassium channel so that they'll leave, because that's going down their concentration gradient to get outside of the cell. So you see that in purple as the action potential is repolarizing, or the cell rather is repolarizing, it's coming back down. Now we mentioned in our previous video that neurons need to be able to communicate with each other, and their connection point is called a synapse, or we're actually saying that they communicate through synaptic transmission. And a synapse is a small gap that's between the presynaptic neuron and the postsynaptic neuron, and it's the way that they're going to communicate with each other. It's important to point out that there are both chemical and mechanical synapses, but the most common type is a chemical synapse. And we're going to focus on that because that's how the neurons communicate specifically with our skeletal muscle. So we're going to use that to talk about how a muscle contraction actually occurs. Because we want to focus on how a muscle contracts, we're going to refer to our presynaptic cell as our alpha motor neuron, and our postsynaptic cell is going to be our muscle fiber. And we're going to have this exchange in communication be electrical when we're in the neuron, it's going to switch to be chemical, and then it's going to go back to an electrical signal within the muscle. So that chemical interlude is referring to these molecules called neurotransmitters. And they're going to be released from the presynaptic membrane, or that alpha motor neuron, and they're going to go or pass through a presynaptic cleft, and they'll bind to specific receptors on the postsynaptic membrane. Remember, in this case, we're saying our postsynaptic membrane is going to be our muscle fiber. When those neurotransmitters bind to that receptor, it's going to cause that cell that those receptors are on to depolarize. And now we're going to get a depolarization of that cell and an action potential that will move through that cell and carry on the signal that we're trying to transmit. As a quick review, what's the sodium-potassium pump doing within the cell? Choose the best option. If you chose that it's pushing 3 sodium ions out of the cell and putting 2 potassium ions into the cell, then you're correct. It's helping achieve that resting membrane potential with a high concentration of potassium in the cell and a high concentration of sodium outside the cell.